Okay, so hi everyone, my name's Tim Galvin, and I'll be talking to you today about how I applied PINK to some radio galaxy zoo data. Something that Ray touched on already was we are entering this new era of radio astronomy. As we have SKA and its pathfinder instruments start to come online, we are going to be probing a new volume of the universe. And up on screen I just show a couple instruments that we are currently in some stage of constructing in Western Australia. So you have ASCAP, you have MWA, and eventually we'll have the low array of SKA. And a project that is particularly close to me and particularly close to Ray is EMU. I was going to talk more about it, but seeing as Ray has already done it, I'm not going to. So I'll jump this slide. But all you really need to know is that it's going to be an awesome survey probing a completely new volume of the... Okay, I'm just hanging on. I don't know what that was, but okay. All right, fine. Okay, yep, so EMU's awesome. I'll keep going. The problem is, I feel like I've missed it. Oh, yeah, so EMU is a radio survey, and it's going to come up with roughly 70 million objects. And the problem we have is how can we cross-catalog these objects with different wavelengths? I show you a couple example sort of images up on the right there. The top one is a simple case. The color is infrared, and the contours are radio. And what you can see is in the center, there's a galaxy. There's a hint of an infrared component. It's kind of hidden underneath the contours, but they align quite nicely. And that we can say is a single sort of intrinsic source. The bottom one is a more complicated example. You have not only sort of a contour and an infrared component in the center, but you also have these two sort of components on the outskirts of the image, those radio lobes. A big problem we'll have is how do we, first of all, recognize that those lobes, those radio contours, are related to a single intrinsic source, and how can we then cross-catalog that with a different wavelength, say infrared, for example? It's important to do that because we need to be able to understand, for example, two radio objects, are they intrinsically related to one another, or are they just by chance close to one another? And often you need multi-wavelength information to do that. With 70 million objects, there's not going to be enough students, so machine learning is an attractive solution to the problem. I was going to try to push the button, but I'm not going to. Self-organizing maps are particularly interesting to me because it's a completely unsupervised method of machine learning. This is something that Kai spoke about yesterday, so I won't spend too much time on it, but all you really need to know is you have a set of neurons. You find from your training data a representative neuron. And not only do you update the weights of that neuron, but you also sort of apply them to surrounding sort of neurons as well. So eventually you have this map that converges to a state that is representative of all features in your, or should be representative of all features in your training data. With PINK and images, that's a little bit more difficult. You have this problem where if you take an object and you flip it or spin it, the traditional SOM would have no chance of recognizing that. That is a single intrinsic shape just rotated. So PINK brute forces the problem, comes up with many different realizations and compares them all. And it does a really good job. Again, this is a poster that Kai showed off yesterday. It was trained on roughly 200,000 images from the first survey, and you can see point sources in one corner of the map, you see complex sources in the other, and a smooth gradual change between the two. You could also go ahead and I should have jumped to this slide earlier, but yeah, you can quite easily see the doubles, the extenders, the weaks, and so on. So it's a really good tool for recognizing different morphologies in your data. A question I wanted to ask is, okay, radio morphologies are cool, but we really need multi-wavelength information to really make sense of what we're looking at. Those doubles, are they doubles because they are radio lobes of an AGN, or are they just close to one another? They could be unrelated star-forming galaxies in close proximity to one another. And you really need multi-wavelength information to do that, to make that distinction. So, okay, let's take something like infrared and see if we can train a self-organizing map that is basically aware of these multi-channels. So think of a JPEG image which might have an R channel, a green channel, and a blue channel. We can do the same thing but with wavelength, multi-wavelength. And can those self-organizing maps be physically meaningful? And how could they potentially be used? So naturally, for that reason, I was drawn to the Radio Galaxy Zoo dataset. It's due for data release number 1, hopefully this year. But the basic idea is simple. It's a citizen science project where users will identify radio components associated with a single source and the corresponding infrared host of that source. The data set for every particular subject will come up with the number of radio components associated with that source, their location, the number of peaks inside each component, so whether, if you think of like a gradient map of the brightness across how many points of inflection there are. And you also get a consensus level, which I think broadly you could say is a complexity measure. And that is simply the agreement of participants who classified a particular subject. Did they all agree that this is a two-component source or a one-component source? Did they all agree where the infrared host was, and so forth? So using their data, I had roughly 100,000 radio components that I could download images of. Each radio component, it also listed whether there were other related components in the catalog to form a single intrinsic source. But basically, each source had at least one component, and each component had at least one peak. I downloaded first data of these these positions from their catalog, and I applied very basic pre-processing. Because this is a radio continuum image, the pixel noise is correlated. Sorry, the noise among pixels is correlated. And you also have these artifacts. You might be able to make out a very hint of them in the left picture. So we applied some very basic pre-processing steps where we sigma clipped the background. We also replaced empty or NaN pixels. We logged and then normalized it under the 0 to 1 scale. So that was the radio channel of these image cubes I made up. I then went and downloaded WISE images of the same position. Thankfully, the resolutions were fairly well matched, but there was a slight derotation I had to do to align the pixels correctly in terms of position. And we performed a similar process, but importantly, we didn't apply the background clipping. I'm not an infrared dude by any stretch, but I understand the sensors are more independent in that sense, so there is far less correlation among pixels. So really all we did there was replaced empty or NaN pixels. We then logged and normalized the data. And you see on the far right there that it's a lot more confused. There are many more sources in the field. And that's something I just want to highlight that now. So in a few slides' time, you'll see something cool. I've got 7 minutes too. So this is one of the self-organizing maps I came up with. The first channel is the radio. You can see quite clearly doubles and singles and whatnot. You can also see some cells where you have two objects. And if I jump to the second channel, the infrared channel, you can see that we've been able to sort of come up with a location of where the infrared host is. And importantly, if I jump ship again to the next page, just some points of interest, you see increasing complexity in terms of the radio galaxy zoo labels. So on the left column is the first, on the right is the WISE. And you can see that we've managed to recover a potential position of the infrared component of these types of objects. But also on the right, we've identified a set of grids, a set of cells where there are two radio components, each of which have their own infrared component. So we can start to make a pretty good guess that these are just unrelated objects in close proximity to one another, if you were to interpret those neurons directly. This should be going to the next page. If we were to take the radio galaxy zoo labels for a set of data and distribute them across the SOM, you can come up with almost like a genome or DNA sequence. This is a zoomed up picture of the lower leftmost cell, and in this particular case, the radio galaxy zoo sources which were classified as having two components, and within those two components 3 peaks, it's much more strongly active. You can see that it changes across the surface of the self-organizing map, and you can sort of see that there are regions of ambiguity where, for instance, if you look at column number 4, we go from 1_2, we start to see sort of 2 activations until we get to 2_2. So that's really cool. You can also use this as almost like a dimension reduction technique. So I've taken 3 sort of sources on the left here. The top row is the radio, the second row is the infrared, and the third is sort of a measure of how similar that source was to each neuron on the self-organizing map surface. So I've gone from 2 channels of roughly 200 by 200 pixels down to a single matrix of 15 by 15 floating point values. And that should, in principle, be an excellent tool to classify with. So with that very small sort of similarity matrix, which I'll call it, we elected to train a random forest regressor. I think I've seen this picture on one of the posters outside, but the idea is you just have a decision tree that compares features that you provide, and it eventually makes decisions until it gets down to some type of classification. And not only do you train one of these, but you train many of them. So we did that, and basically we trained two of them. One of them to try to estimate the number of components in an image, and another to try to estimate the number of peaks. The plots I show up on the right here are basically showing the difference between those two values. If you were to take the radio galaxy zoo data as ground truth, which might not necessarily be a good thing because inherently they are uncertain. They have a consensus level. And the data that I selected to run this test with, I set a minimum of 60%, so 60% of participants had to agree on an answer. So you can see that there's always going to be sort of some difficulty. But if we correctly predicted what the component was or the number of peaks was, we would be centered at 0. And you see that there's a very nice peak there. So we're doing something right. If you were to sum up all the counts inside those dotted lines, you could consider that to be a correct answer. And we find that, you know, we get 75% correct, like if you were to take the RGZ labels as ground truth. You can then go ahead and supplement that information with other information. So I've done the exact same experiment, except I provided the consensus label as an additional feature. So I gave the random forest regressor 226 items and ran the same test, and you see it does an excellent job. Here we're getting around about 85%. The bottom we're still sort of floating around at 74%. But what's important is the sort of ambiguity. We're able to measure whereabouts the uncertainty is coming from. Is it the number of peaks or is it the number of components? You could say you see an image and yes, there's one component, but it's sort of starting to show hints of there being two components. And you can sort of start to capture that in these types of plots. Ambiguity is very important. Just to prove to ourselves that we were doing something right, I ran the same test, but with the images that were provided to Pink directly. So I gave it the 2-channel 200 by 200 pixels, and you see that, yes, it sometimes does a good job, but the spread is a lot more. And you're also, in the number of predicted peaks, you're seeing this sort of structure that we didn't really see before. Even if I was to put the previous plots on sort of a log scale, it was very well behaved. But what's very interesting is if you sum up the number of items in that dotted region, you would find that there was about 75% correct. But you can see that the spread is a lot larger. If you were instead to take the sort of absolute distance from 0 in both of these plots, they are about 30% higher than the ones I showed on the previous slides. Okay, so if you were just to take this hard classification scheme, you would say, okay, training it on the images directly does about the same. It really is sort of hiding this sort of uncertainty that you're having. I've got a minute left. So the next stage of the project is, okay, training it on multi-wavelength cubes. It does a good job. We should be able to use the trained neurons as a predictive tool, well, as a classificative tool. So rather than having to have 100,000 images that I give off to all you people and say, please do 20,000 of them, and then we combine all the answers. In principle, we can train a self-organizing map. We can distribute those 225 objects out, and then we can use it as a tool to sort of transfer the knowledge back. Alternatively, you could classify a collection of sources that went into the training set, like we've done with Radio Galaxy Zoo, and use that similarity measure as a way to transfer that knowledge. You can start to use the trained neurons as the basis of cross-cataloging tools. So there's a number of papers out in the past 5 years where people are attempting to use almost like a Bayesian method of cross-cataloging components to different wavelengths. And we're able to use this as a really nice way to come up with priors. So how often objects that are unrelated are within 5, or 10 or 20 arc seconds of one another. How often is the infrared host in the middle, and so forth. You can use outlier detection. If something doesn't fit onto your map, that's probably something interesting, and you would see that in your similarity matrix, that there wouldn't really be a nice sort of neuron that it matches up with. So, yep, conclusions. I've only got 10 seconds, so I won't go there. But the next slide has pictures and movies, and hopefully they work. Yeah, so thank you.